Apa? Lanit, linknya tuh ternyata nggak bisa dibuka. UGM linknya tuh ternyata nggak bisa dibuka. UGM linkmu tuh ternyata nggak bisa dibuka. Oh ya?
Ini linknya sama kah, Mbak Nita?
Hello, everyone.
Hey, Anais.
Hi, Budi. Nice to see you.
Yes. Did the link works for you or?
Yes, the one that your colleagues just sent me some minutes ago.
Okay.
It's this one that I used. I just shared it with my colleague Anna as well.
That's great.
She will join soon, I assume.
Hello, good afternoon.
Good
Good
afternoon,
afternoon, Prof. Kharis.
Prof. Kharis.
Hi, good afternoon.
Hello, good afternoon. Could you hear my voice?
Yes.
Yes, quite clear, Prof. Kharis.
Nice. Okay, thank you very much. Good afternoon, Anais. How are you?
I'm very good, thanks. I'm very happy to be online today and to meet you.
Nice to meet you.
Yes, my colleague Anna will join soon, and then I think we can. So we plan to present ourselves during the presentation. But so briefly, I'm Anais Glatard. I'm a consultant at Pharmetheus. In Sweden, I'm working from Uppsala in Sweden. And my colleague Anna is working from France.
Yes, hello. I also joined. Do you hear me well?
Yes, Anna.
Thank you. Nice to meet you.
So good. We are in here in Indonesia. Good afternoon. I'm not really sure with you guys. Probably good morning or good evening. So let me introduce myself. My name is Siska. I will open this webinar topic this is the first webinar of the pharmacometric modelings, the introduction and also application. So before the opening, we will open from the opening remarks. It's supposed to be from the Dean of the Faculty of Pharmacy, but I think today he cannot be able to join with us. So the opening remarks will be from the Vice Dean of the Faculty of Pharmacy, Mr. Nanang Fahrudin. Can you give some 2 or 3 words? Thank you.
Okay, thank you, Mbak Siska. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon and good morning, everyone. The honorable guest speaker for today's webinar, Ms. Budi Octasari Susanto, PhD candidate from Uppsala University, Ms. Anna Chan Kwong, and Ms. Anais Glatard from Pharmetheus. And I'm also greeting Prof. Akhmad Kharis Nugroho as moderator. First, I would like to use this opportunity to thank to all of the speakers today for the time they spent and also for the willingness to give a webinar today in the precious topic entitled Pharmacometric Modeling, Introduction and Application. On behalf of Faculty of Pharmacy, Universitas Gadjah Mada, I would like to send very warm greeting to all of you, the participants in this webinar. It is truly an honor for me to be here in this interesting lecture, surrounded by a brilliant mind and passionate individual in the field of pharmacy, especially in pharmacometric study. I wish you all a meaningful and enjoyable lecture and webinar, and please use this opportunity to learn from the speaker. And lastly, but not the least, I would like also to express my profound appreciation to the committee who have prepared this event very well. Again, we really sorry that our dean could not attend this meeting because he has another agenda in another area. And I really would like to apologize that I could not attend this lecture to the end because in 25 minutes I will have a lecture for the student. Thank you so much, and I wish you all an exciting lecture and webinar. Thank you, Mbak Siska. Assalamualaikum warahmatullahi wabarakatuh.
Thank you, Mr. Nanang. So before Mr. Nanang left the meeting, can we first taking a picture for all the participants? Can you please open your camera? Mbak Nita bisa minta tolong di foto? Okay, so probably it will be from me. It's okay. So I will take a picture. First one. 1, 2, wait. 1, 2, 3. Hold on. Another one. 1, 2, 3. Okay. So thank you. So now we will go to the next one. The opening remarks from the head of the Center of Excellent Therapeutic Drug Monitoring, Prof. Dr. Apoteker Ahmad Kharis Nugroho. And also he will be our moderator for today. So Prof. Kharis, the time is yours.
Thank you very much for the time. Could you please activate? I would like to share a screen.
Yeah, okay. Can you?
Okay, thank you very much. Assalamualaikum warahmatullahi wabarakatuh. Good morning and good afternoon, ladies and gentlemen. I'm very proud to be here to be the moderator of this webinar series entitled Introduction to the Basic Concept of Pharmacometrics and its Application in Drug Development. This first webinar series in pharmacometrics is organized by the Center of Excellence of Therapeutic Drug Monitoring, Faculty of Pharmacy, Universitas Gadjah Mada, in collaboration with M.Si and Pharmetheus. And I would like to introduce a little bit about this Center of Excellence. It was established since the 1st July 2023, and the background is the Faculty of Pharmacy, University of Gadjah Mada, believe that the implementation of therapeutic drug monitoring in patients, particularly those who use drug with a narrow therapeutic window, also the use of pharmacometric modeling for patient therapy, the development of standardized bioanalysis, research on interaction between drugs and herbs, and also the bioavailability bioequivalent testing to meet individual needs are strategic matters in optimizing drug services to patients. And our Center of Excellence has several activities since July last year. It's about facilitating research on pharmacometrics of levofloxacin and meropenem, also organizing the webinar today in collaboration with Pharmacometric Indonesia and also Pharmetheus. And this year we will also the host of the annual Asian Pharmacometric Network Symposium workshop that will be organized in 19 and 20 November this year. The theme of this meeting will be the strategic roles of pharmacometrics to optimize drug design and therapy. This will be a 2 days hybrid meeting which combine between plenary lectures, invited lectures from speakers from APN board representatives, also student presentations, and also completed with the afternoon workshop. So please write in your agenda about this important plan of the annual Asian Pharmacometric Network Symposium and workshop. Okay, I would like to introduce the speakers for today. The first one is Miss Budi Octasari Susanto. She is currently PhD student in Pharmacometrics at Uppsala University, Sweden, with the focus on development of translational pharmacometric framework to support tuberculosis drug development. She is pharmacist from the University of Gadjah Mada. She studied pharmacometrics since she did her master degree from master program in pharmaceutical modeling at Uppsala University, Sweden. And currently she is the coordinator of Pharmacometrics PMXID Pharmacometrics Indonesia. The second speaker will be Dr. Anna Chan Kwong. He is an MIDD consultant at Pharmetheus since 2021 until present. She has M.Si in biostatistics, PharmD and PhD in pharmacometrics from Aix-Marseille University, France. Her thesis focused on the practical use of prior information in population pharmacokinetic modeling, and she collaborated with both Sanofi Montpellier and the Hospital of Montpellier. In addition, she has clinical experience as a hospital pharmacy resident at University Hospital of Marseille. And the third speaker will be Dr. Anais Glatard. I'm sorry if I'm not correctly spelling your name. She is a consultant at Pharmetheus from 2020 until present. She obtained her pharmacy degree from University of Lion and her PhD in Life Science from the University of Lausanne 2019, focusing on pharmacokinetic pharmacodynamic modeling of psychotropic drugs. During her pharmacy studies, she gained experience in modeling and simulation through short-term projects with academic research group as well as pharmaceutical companies like Xavier, Glaxo, and also Novartis. And the agenda for today, we will have 3 speakers. The first 10 minutes will be for Ms. Budi Octasari Susanto. She will introduce about community pharmacometric Indonesia. Ms. Budi, time is yours.
Thank you. I will try to share my screen. Let's see. 1 second. Okay. Do you see my screen in the full screen now?
Yes, yes,
Okay.
quite clear. Okay.
Okay. Okay, thank you very much, Prof. Kharis, for the introduction. And it's my pleasure to be here. And also thank you for the organizers for organizing this webinar. And we are very grateful to be able to collaborate with the Faculty of Pharmacy Universitas Gadjah Mada. And it's very nice that I will also be able to come back to my alma mater and be part of this webinar. So today I'm going to introduce you a little bit about our community, PMXID, which stands for Pharmacometric Indonesia. And so let me start. So it started when I studied pharmacometric and start to attend some conference in different countries. But the thing is that it's very difficult for me to find people from Indonesia doing pharmacometric. In fact, that we are the 4th biggest population in the world, and it's very, very difficult to actually meet people that are currently doing research in pharmacometric. So that's how I start to actually build this community. So I start to reach out to some people, and then finally I find some. And then coming also from the mutual connection, I got introduced to some people. Then I started to organize a Zoom meeting almost 2 years ago with some of these people in the picture. And we kind of start officially this community. And then the goal of this community is that we want to gather all Indonesians who are currently studying or at least interested to learn about pharmacometric. And we also hope that this community can provide a space to discuss, to share the knowledge and experience in the field of pharmacometric, because I found that studying pharmacometric is not easy. Basically, it's not an easy subject. So it's going to be very nice if I can actually discuss with someone that is speaking the same language as me. And in the end, we also want to introduce pharmacometric to broader audience in Indonesia, which hopefully more and more people can learn more about this field, and hopefully it will grow bigger in Indonesia. So currently we are around 15 people. Mainly we are coming from the pharmacy and the medicine background. Some of us are studying for our PhD in different university in the world. Some of us are lecturer in different university in Indonesia. We also have some postdoc and specialists working at the hospital, and also scientists working at the company. So yeah, we are quite diverse, and even though we are mainly coming from the medicine and pharmacy background, I've seen that in pharmacometric fields, people can come from chemical engineering, applied mathematics, and statistics. So this field is more like interdisciplinary field. So I think it also opened the collaboration with other fields as well. Initially, for the activity, we are mainly focusing on our community. So we started the activity on sharing the information about PhD position, postdoc, different courses and conference in pharmacometric to encourage people to attend or maybe apply to study in the field of pharmacometric, and to try to increase the presence of Indonesian in different conferences in pharmacometric. And we also have a monthly meeting where we have internal sharing session where people can discuss their upcoming, ongoing, or completed project. So then we can basically learn from each other to get the feedback about our research, or maybe be inspired on our future project. And we have covered different topics such as PBPK modeling, and we also have someone that shared their teaching experience on how they introduce pharmacometric in the undergraduate study. We also cover different disease areas such as TB, HIV, and malaria. And if someone doesn't have any project to present, we can also discuss a paper as a journal club. And for those that just attended courses or conference, they can also give update on what was discussed during the course or conference, so then everyone can be updated and understand what is the currently the hot topic in the field of pharmacometrics. And then after it has been done for like around 1.5 years, we start to go beyond our community and start to introduce pharmacometric to outside our community. And we organized a webinar before. So it was our first webinar was organized in December last year, and it was very successful. And we were collaborating with Universitas Atmajaya to organize this webinar, and it was focusing on how the application of pharmacometric in the clinical practice. And now we are in our second webinar, where we collaborate with Universitas Gadjah, Mada, and Pharmetheus, and we are very grateful for this collaboration. And we will focusing more on the how pharmacometric can be implemented in the drug development. So we are planning also our next webinar, which is coming soon, and we are collaborating with Universitas Surabaya, which will focus on the topic of physiology-based pharmacokinetic or PBPK modeling, and we will have Jeffrey, who is working at Simulation Plus, to give a talk in this. So if you're interested, just stay tuned to our social media account, and we will publish the information when it's official. And also, we are also thinking about our future webinar. So we have been thinking about several topics, such as the application of pharmacometric for bioequivalent study, the model-informed precision dosing, how pharmacometric from the regulatory perspective, also application of AI or machine learning in drug development, because right now we have seen that more and more people are trying to use machine learning in the drug development and maybe combine it also with pharmacometric. We're also thinking about some webinar about the career talk, how it feels like being a pharmacometrician in different sectors like industry, consulting, regulatory, and the clinic, and also some webinar about study abroad within pharmacometric field. But this we also need maybe help. So we are currently looking for a host. So if any one of you is interested to be the host for our future webinar, please reach out to us and we can discuss about the future collaboration. And we are also thinking about organizing an on-site workshop, which mainly focusing on the basic pharmacometric with the hands-on exercise. But it's maybe not gonna be coming soon, but hopefully it can be done next year. Yeah, and also if anyone has a topic that they are interested in, you can also reach out to us. What else can we provide? So we can also help to bridge collaboration between Indonesian institutions to different pharmacometric expert abroad. So in case you were trying to, let's say, start research in pharmacometric and not really sure how to start, etc., and want to discuss with people that are more experienced, then we can provide some help to bridge you to like any expert in pharmacometric abroad. And we can also give some advice on if, for example, the undergrad student want to continue their study in pharmacometric field abroad, then we can suggest like which university and then which program, because sometimes different university also have different focus. Some of them are more clinical, some of them are more on drug development or methodology. And also it can be also that they are quite specific in the disease area. Lastly, so if you're interested to learn more about pharmacometric, I really encourage you to join us in PMXIT, and you can reach out to us in all of these contact listed. So we have emails, we have Instagram account, a LinkedIn account, and also we have the YouTube channel where we often upload our webinars. So thank you.
Thank you very much for the brief introduction about Pharmacometric Indonesia. So I do hope that all participants already write down in their notes about how to join the Pharmacometric Indonesia. Okay, so then we go through the next agenda. It will be the, sorry. It will be the presentation from Dr. Anna Chan Kwong and Dr. Anais Glatard from Pharmetheus, who will talk about overview of pharmacometric modeling in drug development, PK/PD principle, also model-informed drug development, case study in oncology, and also career path and job opportunities in this field. Dr. Anna and Dr. Anais, time is yours.
Thank you very much. I'm Anais, and I will start the presentation. Let me share my screen. Yes. Okay, let me go to the slideshow. Yes, okay. Okay, you can see the presentation now?
Yes.
Perfect.
Yes.
So, perfect.
And
So,
yes, okay.
yeah. So yes, I will start, and I will start by saying that it's a real pleasure to be online with you this morning. For us in Europe, it's 20:09. Since I met Budi, it was really a wish to be able to participate to this webinar with you, and then it took some time to organize a format and the topic, etc. But we are with Anna, my colleague, we are happy to be here today, and I can also say that I was really impressed from the beginning about your enthusiasm, Budi, and your energy that you have in building a pharmacometrics community in Indonesia. And I think it's really important in this field to be able to communicate together, to share. It's really important to learn and to have better knowledge for the projects. So let's start now, and so we will talk about pharmacometrics in drug development, basic concepts and applications. So who I am? I've been already presented. So maybe what I can emphasize is that I've done some pharmacy studies in Lyon, in France, And I had the opportunity also to do a pharmacy residency, where I started at hospital, but I also had the opportunity to have temporary positions, so 6 months positions in pharmaceutical industries and other academic research units. And it's when I got some education and again experience in modeling and simulations. And after that, so I decided to do a PhD. So it was at the University Hospital of Lausanne in Switzerland. And the topic was PK/PD modeling of side effects of psychotropic drugs that are already in the market and used in clinics. And the clinicians were interested in modeling the side effect like weight gain or hyperprolactinemia that are quite common with psychotropic drugs. So yes, so our talk today will focus on drug development as it is the core of our activity in Pharmetheus. But of course, there are plenty of opportunities for applications of modeling and simulations in hospital. Then I will give you the word, Anna.
Thank you. Thanks to all of you who organized this webinar. I also believe it's very interesting to exchange in this community because you will see that it's another language to learn. So it's super nice that you are introduced like this with PMXID, I think. So I also have been well introduced by Ahmad Kharis, but here I can also say that I am a pharmacist as well. We can come from different fields, but we are many from pharmacy. And I also did a residency in France, in Marseille. And there I also did some statistician masters. And during that time, I was doing duties on call in the pharmacology department. So I was doing the therapeutic drug monitoring. And so, for example, we received some blood samples with antibiotics to determine their concentration. And then we could say it's too low, for example, to be efficient, but we didn't use the models. So we couldn't say, okay, you need to increase the dosage by this number so that it's efficient. It was very empirical. So I during that time, I understood that it would be interesting to use the model. And then after this clinical practice, I went for an internship in the industry in Sanofi R&D Montpellier. So it's also in south of France. And I did my PhD during also the end of my residency with the University of Montpellier, the hospital as well, and with Sanofi. My thesis was quite transversal also from development to clinical practice. So I was using data from the hospital and data that I had with Sanofi, because I could use a method, the prior approach, to optimize the models with previous existing models. So that was very interesting for my thesis. And just after, I joined Pharmetheus in Sweden as an model-informed drug development consultant. So I moved in Uppsala, Sweden for 1 year, and now I'm working remotely from south of France, where it's more sunny. Or not during this time, actually, but yeah. So I'm very happy to be in Pharmetheus, and I will present it in the next slide. So Pharmetheus is 12 years old. It was formed by scientists from Uppsala Universitas, and of course with the overall aim to improve patients' lives. So we have to be efficient and deliver with quality, because it's I think an important goal that we have in the end, that the right drug goes with the right dosage to the right patients. Then next slide. This is the map with the Pharmetheus employees' locations. And we are growing, and Pharmetheus is recruiting all around the world. So Pharmacometrics is a field where it's very international, and you can work from home usually. So it's quite interesting for that. And there's cultural exchange with any colleagues, and it's super nice. And then next slide. Our clients are also all around the world, and it's the same. We have to deal with time zone and so on, but it's very interesting. You can take the next slide. This is just to thank the people who we inspired ourselves for the slides, Professor Andrew Hooker and Dr. Elodie Plan from who works at Pharmetheus and Uppsala University, and Dr. Samir Ullah and Svetlana Freiberger, who worked on the presentation as well, and who are colleagues from Pharmetheus. Then you can take the next slide, please, Anais. So the agenda for our presentation with Anais is, I will ask you just 2 questions to see if you would like to participate. It's we would like this presentation to be interactive. So if you have any clarifying questions, you can ask them during the presentation, and otherwise the discussion will be held at the end of the presentation. But you're very, very welcome to ask any questions. We are here for that, so please don't hesitate. So after just asking a question from us, we will present the process of drug development, an introduction to pharmacometrics. Then we will dig a bit into population PK/PD modeling. Anais will talk about the model-informed drug development. We will present 2 case studies from Pharmetheus. And then we will also give you some general information to have things to start with if you're interested to dig more into the population PK/PD modeling and MIDD. And then you will have time to discuss and ask any questions you have. So next. So here is one question that we would like to ask before the presentation. if you know the answer of if the dose of drug A is 100 milligram for a 70 kilos male healthy person, what would be the dose of drug A in a 4-month baby with a weight of 7 kilos? I don't, with Zoom, I think you can write the answer in the chat. A, B, C, D, E.
Or do we have the poll?
All right, we can. I already make it. Wait a minute.
Ah, perfect.
Yes, can you?
Thank you.
Yeah. This is for the question
Great.
number 1, right? Yeah.
Yeah. So I see the second question is also written, so you can
Right.
answer the 2 questions at the same time. So I will let some time, but I'm not sure if I can see the answer. Siska, can you see the answers?
Okay. Yeah. I can, or probably I can make you as the host first. Wait a minute. Can you see it? Anais?
Yes, I can see it.
Somewhere. Yeah.
Okay.
Yeah. And Anna, do you see the answers?
Yes, now I can see the answers,
Yeah.
so I can see that it's
Okay.
very different. So people do not have the same view. But in fact, for both answers, it's we don't know. Otherwise, we wouldn't need some models if the relations were as simple as just calculating based on just these informations. We have to know, for example, for the kidney function, how the drug is eliminated. We have to know as well the relation of its impact on the clearance. And for the pediatrics, it's super, the modeling you will see is super important because in pediatrics, we can't sample the babies a lot. So it's super important to have methods to be able to predict the best dosage. And we cannot really just divide some adult dosage to administer to pediatrics empirically. Yes, so thank you very much for your participation. I hope that this appetizer gives you more insight of why the pharmacometrics modeling is important, because after that, we would be able to answer these kinds of questions, so with more information. So Anais, I will let you present now the process of drug development.
Yes, sure. So I will move the slides, but yes, okay, it's a bit slow. Yes. Let's talk about a process of drug development. So yes, here we start with a drawing and about the magic bullet idea that is the basis of modern pharmacology. And actually, this is about this guy, Paul Ehrlich, which was a German physician and scientist, and he received the Nobel Prize, like 100 years ago, approximately. And actually, he's considered as the founder of the chemotherapy, since he was the first scientist to develop targeted pharmaceutical treatment. And this drawing is about a target that you can see here behind the purple big protein. And this is an illustration that with a drug that is the molecule here with a funny name of magic bullet, you can target a protein which will be activated or inhibited and lead to a pharmacological effect. And here there is a link of this video on YouTube where you can dig more into this idea. But yeah, the two messages is that a drug or a chemical substance could selectively affect the physiological processes. And there are approximately 2 million proteins in the body, but functions of only a few are known. And genes are relatively easier to study than proteins. So with that, when we have about new drug discovery and development, so as you may know, there are different phases. First, we start with in vitro and in vivo testing with some basic research, early discovery, preclinical studies. And this is a period that is quite long in the development, 6.5 years approximately. And usually it's a lot of compounds that are explored at this stage before we go to human testing with a few compounds that are selected. And this is the clinical development with phase 1, 2, and 3, which is also quite long, approximately 7 years. And then it's submission of the new drug application to the medical agencies, like the Food and Drug Administration in United States, like EMA in Europe. And they provide, they review the dossier and provide their answer and hopefully approval. And then it's some post-market monitoring with pharmacovigilance. But this process also for review from the agencies are quite long. I mean, it's a short period comparing to the preclinical and clinical stage, but it's still 1.5 years. So what are the submission trends and the cost of developing new drugs? So on this graph here on the left-hand side, you can see the number of submissions to FDA across year. And we see that it has decreased. Fewer drugs are being submitted to regulatory agencies. But if you look at this right-hand side graph here, which is the cost here in billions dollars for developing a new drug. And here it's before 2000 and after 2000. and the cost has extremely increased. So it's fewer drugs being submitted, but the cost of developing these drugs has skyrocketed. In 2022, bringing a new drug to the market costs $2 billion. So yeah, it's like a picture of what it looks like now. So how we can make the drug development more effective? There are different reasons why a drug does not go to the market. And there are major objections listed here about quality of the drug, about preclinical aspects, clinical efficacy, or clinical safety. And the selection of the dose regimen, here it's 40.5% of the major objections. And I mean, selection of the right dose, it's a milestone in the drug development and a very important application of modeling and simulation. So we can work on that and really improve this by developing models. And also other potential opportunities to have a drug development more effective is to shorten time from discovery to product without risking safety, to stop bad substances earlier because 51% of the substances fail at phase 2, and to ensure sufficient learnings in studies with a more informative clinical trial design. Moving on now to introduction to pharmacometrics, PK/PD modeling. So first, yes, why do we need computer models? So if we have an experiment, here is an example of a car. I mean, yes, it's a real life experiment. It costs something. Here it costs a car. While when we have computer simulations, it's in silico experiments, and we don't use a car from learning. So it's the benefit of simulations, and actually there are a lot of industries that relies on modeling and simulations. So in the field of space, meteorology, mining, defense, cars, energy, telecommunication, aviation, and so on. So it's yes, modeling and simulation are widely used. But coming back to our application in pharmacology. So what is pharmacometrics? So pharmacometrics uses models based on pharmacology, physiology, and disease for quantitative analysis of interactions between drugs and patients. And this involves pharmacokinetics, pharmacodynamics, and disease progression with a focus on populations and variability. So it's really a science that quantify and not qualify. It's really numbers and uncertainty numbers and quantifications that we do with pharmacometrics. And yeah, the aim is to aid efficient drug development, regulatory decision, and a rational drug treatment in patients with application in hospitals and clinics. So PK, it's about drug therapy, plasma concentrations, where we can model the concentrations over time. So we like to say that PK is what the body does to the drug, while PD it's what the drug does to the body. And here it's a representation of the effect as a function of the concentrations, but we can also represent the effect as a function of time. And then we have knowledge about the effects or the side effects over time. So pharmacometrics is the study about effects or side effects, but can also use biomarkers. This is a day-to-day life example about pharmacometrics. So if you think about the coffees that you drink every day, I mean, in Sweden, we are drinking a lot of coffee, but maybe you are drinking more tea, and it's also the same. We can think about tea and tea-in here instead of caffeine. But yes, the idea is to it's to define the concentration over time when you have several coffee intake during the day. And it's an illustration when we can think about drug instead, when the patient is taking a drug at different time during the day. And yes, as Anna said previously, don't hesitate to ask any question if you have some. It's not a problem to interrupt the presentation.
That's fine if there is any question.
No, okay, it doesn't seem. So I will go on. Then pharmacokinetics. So there are different phases. When the drug is given orally, the first phase is the absorption, how the drug will get in the body. Then we have the distribution in the whole body with some transporters that can carry the drug. Then it's the metabolism, how is it broken down, and liver is the main organ of metabolizing drugs. It can also be metabolized in the drug, in the gut. And finally, it is the excretion of drugs, how the drug does leave the body. So the traditional view of pharmacokinetics, we also name this non-compartmental analysis. So it's so in this example, it's an oral dose, and there are 14 concentration measured here, represented by these open dots. And in this non-compartmental analysis, the traditional view, we connect the dots and we determine some parameters like the Cmax and the time at the Cmax, named Tmax. And there is also the area under the curve. And we can also determine the half-life of the drug. But with this traditional approach, missing a data point could easily lead to poor parameters estimation. And we cannot do any predictions for new experiments because we don't have any equation describing this curve. So instead, with the model-based view, the aim is to determine the model, the equation that will describe these points, and we estimate the parameters of this curve. And so in this case, missing a data point could lead to poor estimation parameters, but I mean, the impact is less than in the traditional view, because there are some parts of the curves where if we miss a point, it's less impact on the parameter estimation. Like if, for example, if we miss this point, it will not have a big impact on estimating the parameters of the model. But if we miss a point in the absorption phase or at the Cmax here, it can have a bigger impact on the uncertainty on the parameter estimation. And with the model, then we can do some predictions for a new experiment. So it's really of added value, and it's then applicable in what we call model-informed drug development. So about the PK process and the parameters. So here it's another representation of what I said about absorption, distribution, elimination. And we can have in parallel here the parameters that comes into play in the different phase. So for the absorption, the two important parameters are the bioavailability, which is the extent of the drug that is absorbed, and the rate constant of absorption, Ka. Then about the distribution, we have the volume of distribution. And for elimination, so it's there are quite a large ways of eliminating drug by metabolism, by enzyme, by renal clearance, by biliary clearance as well. So here the parameters is the clearance and the elimination rate constant, Ke. So here it's quite general parameters, but we can, depending on the drug, you can have the renal clearance, the non-renal clearance, depending on the elimination processes. And then about pharmacodynamics. What is the relationship between concentration and effect? So when we draw some blood samples, we can measure the concentrations, and we obtain the concentrations over time. Then about the effect of the drug, so we can, for example, measure a biomarker in the blood and also determine the profile of the effect over time. And when we want to estimate the exposure response relationship, the concentration effect relationship, we will describe what is the effect as a function of the concentration. And I think, and the most widely used model here is the Emax model, which is defined by this curve here. When the effect increases with the concentrations and reach a plateau, which is determined by the Emax parameter. And the equation of this curve is the plasmatic concentration at a certain time point multiplied by the Emax and divided by the concentration at the certain time point plus the EC50. The EC50 is the concentration of the drug where we reached half of the maximum effect. And EC50 is inversely related to the drug potency. It means that if EC50 is very high, we will have less, the drug will have less effect, less potency. But if instead the EC50 is very low, it means that even at low concentration, we will have a higher effect. And the aim of the pharmacotherapy is to lies within a therapeutic window to be able to have the therapeutic effect in the patient. And this therapeutic window is above a minimum effective concentration and below maximum safe concentration. So with this type of representation, we can determine the duration of action, which is when the drug is within the therapeutic window. And what we observed in the two, there are two cases that is most frequent in therapeutic. The first one is that when the maximum concentrations is at the same time of the maximum effect. And then we can have a direct exposure response with here maximum concentration being sorry, the maximum concentration providing the maximum effect. Another case is that the effect is delayed compared to the concentration profile. It means that it takes some time for the drug to have this effect, and the peak of the concentration in plasma is before the peak of effect. And when we represent the effect over the concentration, we can represent what we call a hysteresis curve. So it means that here we have the maximum concentration on this axis, but it's not when we have reached the maximum effect. The maximum effect is reached here. And it's this sort of loop that we observe here that we call hysteresis. And it is the case when we have a maximum effect that comes after the maximum concentration in plasma. And yes, the pharmacodynamics is important to evaluate. Here it's an example with omeprazole, where we have the PK profile here with concentrations over time, over 3 hours here. And we see that the peak of the concentration, it's approximately at half an hour. So it's the Tmax is really short. And here it's a side output, because the action of omeprazole is to inhibit the gastric acid secretion. And we see that here the acid secretion is very quickly inhibited. And then it's the effect is reversing slowly. So yes, that was the first part. Do you have any question at this stage? No, okay, it does not seem so. So then we can move on to population PK/PD modeling, and I give you back the word, Anna.
Yes, thank you, Anais. So here we will have more than one individual profile to analyze, and you will see that we can combine all data to analyze all together and adapt the dosage to special populations, for example. Here on this slide, you can see that we will focus our population PK/PD modeling has a structural model, a statistical model, and covariate model. So the structural model is like the ones that Anais presented. It's like boxes, and we will estimate entrance speed and clearance speed, the volume of distribution. And I will present the different empirical, semi-mechanistic, and physiologically based PK modeling in the next slides, not right after, but in this section. So please, next slide. Can you go to the next slide, please? Here is like the graph that were presented before, but with more individual, and you see that they do not behave exactly the same. You can compute the parameters individually, and then perhaps compute the mean parameters, values, and variabilities. For example, the area under the curve for each individual, and then compute the mean. But then the variabilities will be inflated, and you cannot predict anything with this NCA, non-compartmental analysis type of approach. So next slide, please. If you use population approach, then you will try to fit one model. So like the model that was fitted for an individual, but here for all subjects, while retaining the notion of individual. So you will have this typical profile, population prediction, but you will also estimate the variability around this profile to be able to estimate how the individual would be compared to this typical profile. Yes, you can go to the next slide. Here is the example that you had, for example, in the appetizer. It's a PK concentration of a drug in premature babies. And you can see that I didn't lie. The sampling for babies are super sparse. It's only like 2 points per subject. So you cannot drive any conclusion from non-compartmental analysis. So the population analysis will be able to draw a typical profile and the variability around this from this data. And then it will be possible to use this model to predict, for example, how to what is the best dosage for the next premature baby who will need this drug. So next slide, please. This is just the schematic of drawing that summarize what I said. With the same observations, you can use the traditional methods or in silico methods, so the modeling, and then you can predict the future with the models. Whereas the traditional approach, you can have parameter estimates, but not to use for prediction. Next slide, please. So I told you the population PKPD model compartments are structural, statistical, and there's a covariate model that we can search for how patient characteristics will influence the PK or the PD, depending on the model. Next, please. The structural model, it can be empirical, semi-mechanistic, or physiologically based. So the last one is a kind of a different branch. When we talk about POP-PK and POP-PK-PD, usually it's the two first models. We represent the body in boxes, and then we will estimate some parameters to describe how the drug enters and is cleared from these boxes and is distributed as well. The difference between empirical and semi-mechanistic is that when it's semi-mechanistic, we will try to use the knowledge we have on the body and the drug to be able to assign the boxes to, for example, the lungs, and to use some of the boxes yes, in a mechanistic way. And the PBPK, it's the same approach with boxes, but here we will have the knowledge that we have for the entrance and for all parameters before having the data to be able to predict the data. So next, please. Oh, sorry, it was this schematic of PBPK with the every box as one organ. So here, next, please, for what is interesting in population PKPD, in fact, is the statistical model that is to estimate the variability around the typical parameters and the typical profile. You can estimate variability between individuals and within individuals. So you can take the next to have the graphic, perhaps. So this is, for example, the typical concentration profile versus time. And if you click next, you see for one individual, you will have a residual error. And then if you click next, yes, and for every individual, for all the individual present in the dataset, you will have not the same typical profile, and you can estimate the between subject variability. And next, please. And the last facet of the population PKPD models, it's the covariate search. We include, or we try to search for covariate relationships to explain between subject variability, because sometimes the subjects are very different, but it's because of a characteristic that we can measure. So it's super interesting to determine what a covariate drives or have an impact on the model. So, for example, if it's the creatinine clearance, as we told you in the appetizer, if we know that the creatinine clearance can impact the pharmacokinetic of the drug, we would test it and know what is the relation. For example, it wouldn't be please divide the dosage per 2 because it's the creatinine clearance is increased by 2, but it can be another relationship. So including this covariate in the model would increase the predictive performance of the model. And then you can also directly adapt the dosage to some population subgroup. So here, for example, in this example, it was the renally impaired patients. And of course, it's better if the covariate can be explained mechanistically. So, for example, if the drug is cleared by the kidney, then it's super. We would be confident that this covariate is in the model. When to include covariate relationships. When indicated by some goodness of fit criteria. So, for example, if you stratify your patient population on your covariate, and then it's totally different profile. It's good to investigate this, and when it increases the usefulness of the model, of course. So we will try to identify covariates that we can measure. For example, if it's for the clinical practice, we will need covariates that we can have at the beginning of the treatment instauration. So next, please. Here it's the example of an antibiotic, the gentamicin, used. It's still in premature infants, so to treat pneumonia. So it's efficient, but it can also be toxic. And I told you about the therapeutic drug, the window. So here it's a narrow window between peak and trough target exposure. So we need to adjust individually the dose between effect and toxicity. And some scientists build a model so that they could determine which dose would be appropriate in neonates or even in premature babies. So next, please. So the data they had to build the model were only sparse. It was on premature babies, so difficult and non-ethical to sample a lot. There were many different doses due to heterogeneous population. So yes, perhaps it was adapted, the dose empirically based on age, weight, kidney function, and the concentrations were and the dose were changing dramatically with time. So it's difficult to predict if you have very different profiles, PK profiles, and then you want to predict which dosage you want to administer to the next premature baby. How do you do? So it's better to, so it's when the modeling is useful. And if you take the next slide, they the scientists in this publication used a one compartment model with linear elimination. So it's like the one compartment model that Anais presented. And then they search for covariates. So you see that weight impacted the PK parameters, and there were quite some variability on the PK profile, but they could estimate the impact of some of the covariates and explain 40% of the variability between individuals by including the covariates kidney function, age, and APGAR score. It's the health score for premature babies. If you take the next slide, here you have the reference of the article, and you can, in the article, they just recommend to increase the dosage, to be efficient and still non-toxic overall. And yes, it's important in clinical practice. We will talk about some other example later in drug development. So please, if you have any questions before we move to model-informed drug development with Anais, you can ask them. Then we can move on.
Yes. Thank you, Anna. So now it's time to talk about model-informed drug development, MIDD. So yes, it's always a bit slow when if I mute or unmute myself for the first slide, but now we are here. So model-informed drug development, so it's defined as a quantitative framework that aimed at improving the quality, efficiency, and cost effectiveness of decision making. And so here it's 3 papers that were the basis of model-based drug development. And this one is about a rational approach to efficiently accelerate drug development. And this one is CPT-PSP. It's a white paper about the good practices in MIDD, or yeah, you can find different names for MIDD. Here it's model-informed drug discovery and development, and it's why it was abbreviated MID3. So yes, it's different wording. So, but yeah, the concept is to use modeling to inform drug discovery and development. And so there are different authors in this paper, but there are three colleagues at Pharmetheus with Peter Milligan, our CEO, Richard Anziano, our principal consultant and chief statistician, and Marie-Laure Chenel, who is chief research officer now. So they are co-authors of these papers, and I think it's good ones to read to dig into MIDD. And here is a table summarizing phases of clinical development and what is the aim at each phase and what are the data collected. And so actually, yeah, the aim of MIDD is to use the data collected from the very first phase, and with this to be able to leverage this data with models and to inform, for example, the dose selection in phase 2 or some study design of studies in phase 2 or phase 3. So yes, this is some example of what we can do for MIDD. And at the beginning in phase 1, the aim is exploratory about safety and tolerability. It's a few subjects involved in the studies, and the data collected, it's about adverse events, PK parameters, sometimes PD measure, but the PD will be evaluated much more during phase 2 and phase 3. Phase 2, we start with preliminary safety and efficacy to support go, no-go decision to later phase in the development. It's the first studies including patients, and we will have the preliminary evidence of efficacy, what we called proof of concept. In phase 2B, it's the start of confirmatory studies with dose selection to support the registration of the drug. So it's where with models we can really determine the best dose to use depending on efficacy criteria and also safety criteria. So we can develop PK/PD models with an efficacy biomarker, a safety biomarker, and we determine the best dose regarding efficacy, the best dose regarding safety, and then we inform the decision on which dose to select. And then in phase 3 is again confirmatory again. It's not the same because here it's wider studies with much more subjects. So it's about efficacy and safety to support the registration. And we get more confidence about the right dose to use for the drug approval. And then in phase 4, so it's post-marketing surveillance that is mandatory to reveal unexpected adverse effects or toxicity. And this includes treated patients, and the data collected are about adverse events. And there is regulatory encouragement from FDA in the US, EMA in Europe, but also PMDA in Japan about MIDD. And here it's yes, some good publication to read if you want to know more. So here we provide some numbers about how MIDD increased efficiency. So it's 25 years since the discipline of pharmacometrics in drug development started, and provided a different shape of drug development comparing to before. It's 84% of FDA approvals that had labeling decisions informed by MIDD. You can know more in the first publication provided below. And it's 12% projected annual growth rate in the overall biosimulation market. And some achievements. So it's approximately 2,750 patients fewer in the trial due to optimization or of study design. I will show an example later. It's also some time reduction of drug development. So 2 years shorter program. And also some cost reduction, 50 million US dollars saved in research and development costs. So if you remember at the beginning of the presentation, we showed some numbers about the time and cost of drug development. And here it's how MIDD could reduce these numbers. And yes, I encourage you to go to this publication if you want to know more. Yes, so a little bit more details how MIDD can support the drug development. Some important decisions during drug development. So it's about formulations and dosing frequency, about the duration of treatment, some study success criteria, about the endpoint of interest to evaluate for efficacy. For example, usually there is a primary endpoint to determine efficacy and some secondary endpoints. MIDD can also support dose, sequence of administration, schedule of administration, combinations of drug, about particular populations like premature neonates in the example that Anna explained. It can be also in renal impaired populations, in elderly populations. It can also support some indication characteristic and our competitive landscape. So it's when a drug can be used for different indication. So we evaluate, which is the one that has more probability of success, which indication is the most appropriate for a drug. And finally, about differentiation or transition criteria through research development of our registration and reimbursement. So it's also some aspect of pharmacoeconomy. And here it's how MIDD can reduce the number of subjects needed in a trial. So once you have a model available for a drug, you can determine with optimal design strategy when it's the most informative to draw the blood sampling for the biomarker, for example, that you would like to study. And once you have a more informative design and optimized design, so in terms of sampling times, then actually you can have more power in your statistical analysis, because at each sample time you provide more information to develop the model or to analyze the covariates. And for example, here it's a traditional design compared to the optimal design, and to reach 80% of power with optimal design, we need a little bit more than 10 subjects per treatment group, while you need 40 for a traditional design. So it's really a gain of using human data. I mean, the less subject we involve in study, the more efficient we are. It's also more cost effective. And here it's, I won't go through into each details because I already talked about this, but it's another view, another way of showing how MIDD can inform the drug development. So it's across different phases, from the target selection and validation up until the approval phase. And it's, again, some different example where MIDD can impact, but which is also maybe interesting to see here is that at the beginning, it's more a support for internal decision. And when we move on towards the approval phase of the drug, MIDD supports more the regulatory decision. So which impact can be expected or may be anticipated with MIDD? So it's where we can have low impact, medium impact, or high impact. And so this is impact on internal decision. like on the dose selection, for example. And here, so it's EMA that classified this impact with describe, inform, and replace. I will explain this a little bit more. And this was nicely illustrated by the EFPIA here, European Federation of Pharmaceutical Industries and Associations. So it means that when you have a low category impact, so low impact doesn't mean low value here, but it's when MIDD provides inference, which has limited impact on internal decisions. So it's more, yeah, a descriptions. And then MIDD can inform and provides inference on and provide inference, which informs internal decisions. And the highest impact, its MIDD approach provides inference, which informs internal decision without requiring additional experimental or trial data to be generated. And there are some cases where I'm thinking about studies in renal impaired subjects that now FDA is recommended to use modeling and modeling application. So it's an example where MIDD can replace some real life studies. There has been recently recommendation guideline from FDA about pharmacokinetics in renal impaired subjects. Yes, so we will move forward with some case studies, unless you have any questions on this late part. No, okay, so Anna, I let you start with this case study.
Sure. So here you can read a case study that we had at Pharmetheus. You can read the principal question. So it was a biosimilarity study on rituximab for the patients with DLBCL. It's the diffuse large B-cell lymphoma. So Dr. Reddy's wanted to commercialize its generic and had to do a study, generic biosimilarity study. So it's a generic versus branded. So the reference project. And they found different PK. So they asked us to model and to confirm if the PK was different because of differences in the drugs, or perhaps because by random chance the population in the two treatment arms were different. So what did we do for the analysis? We analyzed all data together, the reference product and the new product, and then we did the stepwise covariate modeling. So we searched for covariates in this PK analysis, and found that the AUC was lower with a higher tumor size. So you can see it in the figure. And then if the model corrected for this covariate, the drug as covariate was non-significant. So in fact, the difference was due to other covariates in the population and not to the drug. Then a PD analysis was conducted as well. And none of the PD endpoints identified a difference between the drug. Yes, so in this case study, we had difference in the NCA, the non-compartmental analysis, but these differences were understood by the modeling with the covariates. So it was an impact of informing the study for this modeling. Yes, and it was submitted to the agencies. So if you have any questions on this case study, there's an article that is on the reference, or you can ask right now. It was published. So we can go to the next case study from Anais.
Yes, sure. Yeah. Thank you. So the second one is about the CAR T cells used in oncology. So what are CAR T cells? So it's cells from the immune system. Sorry, cells from the immune system, the T cells, that we modified with a chimeric antigen receptor. So it's called CAR with the abbreviation. And this antigen receptor added in the cell will be able to target the cancer cell. So actually, it's in the process of this cell therapy, we collect some T cells from the patients by blood sampling. Then it's an very important steps in laboratory. It's really about engineering and to add so the antigen receptor to the cells, and also to make the cells proliferating. And then when this is mature, the CAR T cells will be injected in the patients, and with the aim to have a therapeutic effect against the cancer cells. The thing is that then it is a principal question of this case study. In absence of clear dose exposure relationship, are there other means to optimize CAR T cell exposure? And the impact of this work was to identify potential MIDD applications for the development of CAR T cells therapies. So what has been done? It's to identify actionable variables which can be modified to optimize the exposure. So it means that we identify where we can, at which stage of the production of the CAR T cells we can optimize the exposure, but also how we can optimize this exposure. And it is explained here that at the R&D stage, So when we define the affinity of the chimeric antigen receptor to target antigen, about the choice of the co-stimulatory domain. So yeah, here it's how the CAR T cells will be stimulated, and also some change in the antigen receptor domains. And then it's when we modify the T cells. So in this manufacturing stage is the treatment of the apheresis from the patient, the conditions of ex vivo expansion of the cells, and the duration of the ex vivo expansion. And finally, at the clinical treatment stage, it's lymphodepleting regimen in the patient and some co-medications that the patient will take to optimize the efficacy. And then how we can support such optimization processes, we identify some suitable mathematical models that are illustrated here with some publication if you want to know more. So I think, yeah, we are approaching the end of the presentation, but we have a little bit more general information, and Anna will tell us more.
Yes, so it's just if you're interested, if you were interested during the presentation, to show you some job and research opportunity. If you want to dig into it, what do you need to begin with? And some links and reference for other sources of learning, but Budi already provided some for you, and I'm sure PMXID is super helpful for that. But if you go to the next slide, you have an article from all, or from a lot of experts in the field. Perhaps if you know a little bit about it, you recognize some names who, so the article talk about training the next generation of pharmacometric modelers. So perhaps it's you. It's a multi-sector perspective. It's not only from pharmacy, but it's you can come from any field, like statistics, for example, or even chemistry. It's and so it can be an interesting read for you. And for the next slide, to work as model-informed drug development consultant, you can work in consulting companies like ours. You can work in pharmaceutical industries. So many have dedicated pharmacometrics departments. Our CRO is only pharmacometrics, and it's lots of colleagues working on this field. And then you can work in academia. Usually to work in this field, you need a PhD. And then you can stay in academia as a postdoctorate, research assistant, research associate, scientist, and take a faculty position. Or you can work as well in regulatory agencies. And next slide. All you need in the beginning is a computer. You need some softwares. I will just list them after. You need data, so you need to collaborate with usually some clinical or industry who has data to work with, and you need expertise, so you can go to seminar or workshops, you can take courses. For example, the 2 weeks summer school in Uppsala when you are in when you are a PhD candidate. and some conferences. And of course, you can use the great initiative of PMX ID, I believe, and attend, for example, the APN meeting. Otherwise, it's a lot of meetings around the world. So if you take the next slide. About the softwares, I told you it's like a different language, so it's nice that you are able to exchange in your own language about the implementation of pharmacometrics, because you will use a lot of coding, for example, in non-mem. That's the software for gold standard for pharmacometrics. There's also Monolix, and R is used a lot. And for PBPK, there's PKCM, SimSip, GastroPlus, etc. If you take the next slide, where to start from? It's nice to refresh your PKPD knowledge as well as your math statistics lessons, and to learn population and nonlinear mixed effect modeling approaches. You have the reference. Usually modelers are super happy to help. You can start learning computer programming. R and RStudio are quite used in this field. So you have the reference also in this slide. If you take the next slide, we listed other sources of learning with some links that you will have. And that you can access for free, normally, for example, on YouTube. So yes, you can go to the next slide. Now it's the end of the presentation. We are very open to questions. And during that time, we will go to the next slide, and that presents, that is just for a survey for us to know your feelings about this presentation. So you can scan the QR code or type the address in your browser while we answer the questions that you hopefully have. Thank you.
Thank you, Anna and Anais, for a very clear and nice presentation. In the chat box, there are already several questions. The first one from Eri Prastio Nugroho. He asked about what kind of product that Pharmetheus offer. Is it a software or application like other companies such as GastroPlus, SimKeep, etc.? And also, is there any internship opportunity in Pharmetheus for students?
So maybe I can answer. For now, Pharmetheus is not provided any software. And what we offer is services of pharmacometrics analysis, of MIDD applications, of different type of models, some data programming, of course, because we need some in order to have an analysis data set used for non-mem, for example. And we can provide some, the type of work that we provide is either really a consultancy work, where we have a contract between our clients and us, and we do the analysis on the Pharmetheus side, if I can say, but we can also have other type of contracts where we work fully for one client, and then it's not for a specific analysis, but it's maybe for a day-to-day question of pharmacometrics. So we are more embedded at the client side.
Okay.
And yes, and then the other question was about internships. Yeah, so yeah, there have been some interns roles at Pharmetheus, and I think if you are interested, you can reach out to the website or to one of us, and we will contact internally the right people.
Okay, nice for the information. And the next question about how far or how common is the machine learning or artificial intelligence application in pharmacometrics in industry practices? This is from Andi Sudiro.
Yes, so it depends what is like what is meant by machine learning. But for example, the stepwise covariate modeling could be considered as one facet of machine learning, and it's more and more into yes, machine learning and AI is more and more used for modeling, but at Pharmetheus we are using like traditional approach, as I can say. Or Anais, perhaps you have a different view.
No, I think you're right. The artificial intelligence methods are growing within the field, but it's still not the gold standard. We still need to understand better what can be the best approach to use this method, the best applications. And so at Pharmetheus, we do not currently use these methods in our work with clients, but we do have some research work about an optimal use of these methods.
Okay. The next question from Amy Octaviani. She is a PhD student in ITB Bandung. Hello, Miss Anna and Miss Anais. I have two questions. The first one, on the PKPD statistical model, is there any program for beginner to use beside non-MEM? And then the second one, how the relationship on TDM with pharmacometric modeling? Thank you.
So for the first one, Monolix is another software to that could be more beginner friendly, but I didn't use this software a lot. And at Pharmetheus we use non-MEM. So, but I know that it's you see a bit less coding in Monolix. And you have nice tutorials on YouTube for Monolix, so it can be a good point to start with. And then on the relationship on TDM with pharmacometric modeling, I'm not sure to answer well to your questions, but if I understand correctly, it's how the pharmacometric modeling can be used in TDM. So in fact, if you receive a sample in therapeutic drug monitoring and you you find the concentration in this sample, then it's too low or if it's too high, how do you adapt the dosage? And using a model could tell you how to adapt the dosage. Otherwise, it's empirical. You will just look at the history of the patient, the history of other patients, how the drug would react when you increase or decrease of some units. And using a model could allow to have a more precise answer and to arrive to your objective, your aimed, your target concentration more rapidly. So did I answer the question, or perhaps Anais, you understood another?
Okay, Miss Annie, if you have any further question, you can just directly ask. There is still some times.
Enough, Prof. Kharis.
Yes,
I
yes.
understood.
Question
Thank
directly.
you, Miss.
Perfect. Thank you.
Okay, thank you very much. Is there any more question from the attendees? If not, I have a short question for both of you. How is the recognition of pharmacometrics method by the regulatory agents so far?
Yes, so I think it's very well received and even recommended. So, for example, FDA has issued some guidelines about population pharmacokinetics and also about renal impaired subjects. So there you can read how, you can read their recommendation on how to perform such POP-PK analysis during drug development. Yes, they are very much well receiving this type of analysis, and as said during the presentation, so it's EMA that classify some MIDD approach with describe, inform, and even replace. So in some circumstances is they are open for model-based simulation to give information for the drug. And I'm thinking about the pediatric investigational plan when it's something that is very important in the drug development. Yeah, obviously not for all drugs. There are some indications when we don't need an pediatric investigation plans, but it's an important step in the drug development, and there the modeling can really help to define a dose in children, and can replace some real studies with children.
Okay, thank you very much for Anna and Anais for a very clear and nice presentation. And time is up now, so then once again, thank you. Also for Budi, I do hope that we can continue our collaboration in the future. And then I will return time to Siska. Thank you.
Yeah, thank you, Prof. Kharis. And I think thank you, Anais, Budi, and Anna for the nice presentation. It's for me, myself, it's new things and it's fun to learn something new. I hope all the participants also feel the same like I felt this afternoon. And thank you very much for the time, and also Anais, Budi, and Anna. As Prof. Haris say, I hope we can continue the collaboration, as this is, I hope, will be the beginning of all the things. Okay, so thank you from me and to all participants. I think we can end this webinar and see you in another opportunities. Thank you.
Thank you very much.
Thanks a lot for this opportunity, and very nice to have to have this time with you. Very good, and thanks a lot for the organization.
You're welcome, Anna and Anais. Budi. See you for the next time. Bye.
Bye.
Thank you.
Thank you.
Bye-bye.
Bye.